Hey grade 10s, welcome to a wonderful Monday morning for me, afternoon for some of you guys, but welcome to yet another day of our science class. Unfortunately, today's going to be a little bit different, but hopefully we're still going to learn nonetheless. So let's get on into things. Today we're going to be talking a little bit about different applications of cellular transport in industry as well as medicine. Now, knowledge about the makeup and functions of the cell membrane and about different methods of transporting substances, which we've been learning about over the past, well, before our break, we learned a little bit about that. But knowledge of those ideas has advanced research in both industry as well as medicine. Efficient natural systems are quite often the model for different types of manufactured systems that humans come up with. And the cell membrane is actually an excellent example of what we call a selective gatekeeper. Knowledge of the cell membrane has prompted industrial use of synthetics to mimic different types of natural functions. And this area of research and development is what we know as membrane technologies. The molecules that are associated with the cell membrane on the surface of the cell are very important for many different types of cell activities. So some proteins, called recognition proteins, are embedded in the cell membrane but stick out into the cell's surroundings. They allow cells to recognize one another. So for example, when an egg and sperm cell link together, or when cells of the human immune system recognize and destroy invading bacteria and viruses and things like that. Those are some very good examples of that. Other types of proteins are what we call receptor proteins, and they may bind specifically with certain molecules to bring them into the cell by endocytosis, which we discussed a couple weeks ago. Some of these proteins have sugar groups attached to them that make the binding specific, and it allows the cell to identify a particular bacteria or a particular virus. In a reverse way that has a negative effect on humans, some viruses, like human immunodeficiency virus or HIV, hepatitis and influenza, they use the binding reaction to target human cells. In other cases, the binding reaction on the surface of the cell acts like a molecular switch, and it can trigger many different activities in the cell. Pharmaceutical research sort of draws on this understanding of the cell membrane and proteins to develop and test new types of drug therapies by making use of the cell's ability, so either that of the disease-causing organisms or of the host, to be able to recognize molecules. And this is one of the reasons why studies of the three-dimensional shape of molecules are really, really important to various health research as being of interest to themselves. Researchers have been working really long and hard to find treatments and cures for constantly changing viruses, so things like HIV. And new discoveries focus on recognition or receptor proteins in human cell membranes that appear to be the attachment point for the virus. So in understanding these proteins, scientists suspect that they may be able to produce a lock and key scenario that would help to prevent the virus from getting into the cell. So the process would kind of work like to block off or close off the receptor protein so that the key produced by the virus isn't able to work. So essentially the virus would be locked out. So kind of imagine the idea of covering the keyhole of a padlock with something like cement. So that even if you had the right key for the lock, you wouldn't be able to use it. Being able to turn away the virus like this would mean that it would not be necessarily to put a whole ton of drugs and things like that into a person's body that could affect healthy cells as well as the infected ones. So the disease could be stopped before it even takes hold. Now, cancer research has also targeted the study of recognition proteins. So common treatments for cancer they're not at this point specifically able to single out only the defective cells. They affect healthy cells as well. If it were possible to identify the unique proteins of cancer cells, it might be possible to develop drugs that are specific to those proteins and therefore specific only to cancer cells. So thinking along those lines, if certain recognition proteins specific to cancer cells could be identified, they could then be used to stimulate the immune system to detect and destroy the cancer. The ability to be specific and target only cells of the cancer would mean you have less overall discomfort for the patient. So that would be a really, really positive thing.
In the late 1970s, scientists discovered an interesting property of phospholipids. That is, when they are hydrated, the hydrophobic tails mutually attract, while the hydrophilic heads seek the aqueous medium, thus forming a phospholipid bilayer around an aqueous core. The lipid bilayers then seal off to form a liposome. Since liposomes are composed of natural lipids which are biodegradable and biologically inert, their toxicity is minimal. The amphotericin B molecule is a good candidate for liposome-based drug delivery because it has a useful duality. On one side, it has a polyhydroxyl chain which is hydrophilic, and on the other side, it has a polyene hydrocarbon chain which is lipophilic, which allows the amphotericin B molecule to be encapsulated between the double lipid layers of a liposome. However, creating a true liposomal preparation is expensive.
While protein research continues, drug therapies also use a structure manufactured to act like the cell membrane. Liposomes are fluid-filled sacs surrounded by a phospholipid bilayer, identical to the cell membrane in human cells. Liposomes were first produced in the early 1960s, and ever since then they've become really important agents in the delivery of drugs to different types of infected body tissue. They're able to be produced for almost any need by manipulating the composition of the membrane. So liposomes are microscopic, so really, really tiny, about one 1,000th the diameter of a human hair. So you can imagine how tiny a human hair is. And these liposomes are only about 1/1000th of that diameter. Water that's trapped on the inside is able to hold water-soluble medications, while the membrane layer is able to hold fat-soluble medications. So the tiny sacs can be introduced into the bloodstream, and they're able to circulate throughout the body. Now, because the membrane is identical to that of the human cell, the liposomes are able to attach to infected cells and deliver the medication that's needed. HIV and cancer therapies really make use of liposomes to be able to deliver medication. The advantage of using these is that the spheres may circulate in the bloodstream for longer than the medication on its own, so that allows for longer sustained treatment. Sometimes liposomes are able to concentrate themselves at the site of a tumor or an infection. And so in that way, they're able to deliver the drug directly to the targeted cells without affecting any of the normal cells surrounding it. The use of liposomes in gene therapy to inject DNA into tumor cells is yet another application of liposomes that's actively being researched. The DNA is contained inside the liposome. A molecule on the liposome surface fits onto certain cancer cells to recognize and target the correct cells. And so in this way, the DNA is able to be introduced into the tumor cells and begin the production of toxins to kill the cell. The only thing is that much more research is actually needed to be able to make sure that healthy cells are not also susceptible to the gene or drug-carrying liposomes.
When you hear the word insulin, You may think of a drug taken by people who have diabetes. While this is true, what you may not know is that insulin is one of the many hormones created in the human body. Insulin is important to the body. It allows blood sugar, or glucose, to get into cells to provide them with energy. When you eat, your body breaks down food into glucose in your small intestine. This is your body's source of energy for everything it does, from working and thinking to exercising and healing. Glucose travels through your bloodstream looking for individual cells that need energy. For glucose to get into the cells, it requires insulin. Insulin is the key that unlocks cells for glucose to enter and deliver energy. When insulin arrives, it signals the cell to activate glucose transporters. These transporters pull glucose through cell walls. When glucose moves into the cell, it delivers energy. Insulin is normally produced in the pancreas by specialized cells called beta cells. When glucose enters your bloodstream, the pancreas matches it with the right amount of insulin to move glucose into your cells. In people with diabetes, this process doesn't work as it should. In type 1 diabetes, scientists believe the body's immune system mistakenly attacks and destroys beta cells in the pancreas. A person with type 1 diabetes loses the ability to produce insulin. In type 2 diabetes, the pancreas is not producing enough insulin to meet the body's needs. Over time, the amount of insulin typically becomes less and less. In some type 2 diabetes patients, cells build up a resistance to insulin. Even though there may be insulin in the bloodstream, it is not enough to unlock cells to allow glucose to enter. As a result, it takes more insulin to find the right key to unlock the cell for glucose. This makes it more difficult for cells to get the energy they need. When glucose can't get into cells, either because there isn't enough insulin or because the body is resisting it, glucose begins to build up in the bloodstream. As a result, all that energy is wasted. It does not get to cells where it is needed. Without glucose in your cells, they lack the energy they require to keep your body working. To keep glucose from building up in the bloodstream, an external supply of insulin may be needed. Because people with type 1 diabetes can't produce their own insulin, they must inject insulin several times every day or receive insulin through an insulin pump. Many people with type 2 diabetes take insulin too. Injected insulin acts on glucose in a similar way to insulin the body would produce if it could. Like the body's insulin, injected insulin helps reduce the amount of glucose in the bloodstream by getting it into cells where it is needed for energy.
Before the process of dialysis was available, people who experienced total kidney failure would end up dying. Today, the availability of dialysis and kidney transplants means that a lot of patients can continue to live full lives. Two types of dialysis are actually possible: peritoneal dialysis and hemodialysis. Both of them are based on the principle of diffusion and osmosis and the operation of concentration gradients. The purpose of dialysis is to rid the blood of toxins, wastes, and even excess fluid that's produced by the cells of the body. So normally, healthy kidneys would perform this task, and it sends waste to be eliminated in the urine. In humans, cells form a membrane called the peritoneum, which lines the abdominal cavity. Now, during dialysis, waste products from the blood pass through these cells into a fluid, the dialysate fluid, and this is termed peritoneal dialysis. A really soft plastic tube, or a catheter, is surgically inserted into the abdominal cavity. The sterile dialysate fluid is pumped into that cavity. The dialysate has a composition that's very similar to human body fluids, and it consists of a mixture of water, glucose, and certain substances that the body needs. Dialysate usually contains sodium, magnesium, chloride, potassium, and calcium salts. The dialysate has no toxins or wastes present, so the concentration of these materials in the blood is much higher than it is in the fluid on the other side of the peritoneal membrane. The movement of toxins and wastes is down the concentration gradient. The wastes diffuse across the membrane and into the dialysate. As the cleansing fluid becomes saturated or full of wastes, it's removed from the body, disposed of, and replaced with fresh dialysate until the entire exchange process is complete. The patient is able to perform peritoneal dialysis at home while carrying on most of their regular activities. Hemodialysis, on the other hand, is actually a much more complicated procedure, and it has to be performed in a health facility. So the blood has to be removed from the body, cleansed using a dialysate fluid in a very special machine, and then returned to the body. The patient isn't able to move around during hemodialysis.
As the Earth's population continues to grow and develop, our limited freshwater resources become increasingly scarce. We are fortunate that the Earth's oceans offer an alternative and can provide a sustainable supply of potable water. Seawater can be economically and reliably converted to potable water through a process known as seawater reverse osmosis. The process starts by extracting water from the ocean using wells located on the shoreline or by using an intake structure located in the open ocean. Osmosis is a naturally occurring process where a solvent, such as water, passes through a semi-permeable barrier. The semi-permeable barrier, or membrane, allows some things to pass through it, but not others. In nature, the direction of flow through the membrane is from a less concentrated solution, such as fresh water, to a more concentrated solution, such as seawater, until equilibrium is reached. Reverse osmosis is when the opposite occurs. By pressurizing the concentrated solution, the seawater, we are able to force water molecules to pass from the salty seawater solution through the membrane to the fresh water. To protect the reverse osmosis membranes from becoming clogged by solid particles that can be suspended in the seawater is filtered before passing through the membranes. This is accomplished by using multimedia filters, which are tanks or vessels containing a series of layered granular materials. These materials can be anthracite, garnet, sand, pebbles, and/or gravel, which are assembled in layers. The filters remove sand, twigs, seaweed, and other particles from the seawater. In some cases, other types of membranes, known as ultrafiltration and microfiltration membranes, are used instead of multimedia filters to pre-treat the seawater. Next, the filtered seawater travels to the cartridge filters, which act as a second stage of filtration. Cartridge filters used for seawater reverse osmosis are typically made from a yarn-like synthetic material that is wound into cartridges. These remove even smaller solid particles from the seawater, such as fine sand and clay, before the seawater proceeds to the reverse osmosis membranes. High pressure pumps increase the pressure of the seawater up to 1,000 psi. The pressure needs to be sufficiently high to overcome the naturally occurring osmotic pressure and force water from the saltwater side through the reverse osmosis membranes to the freshwater side. The salt particles in the seawater are rejected from passing through the membrane to the freshwater side and remain behind on the concentrated saltwater side. The reverse osmosis membrane can be thought of as a number of sealed envelopes connected at their open ends to a tube. There are spacers between each envelope which allow water to flow across the membranes. The membrane envelopes and spacers are then wound around the tube like a roll of paper towels. The reverse osmosis membranes are then enclosed in a fiberglass shell. The membranes are connected end to end, usually 6 to 7 membranes together, and housed in vessels that are built to withstand pressures up to 1200 psi. As the pressurized seawater enters the pressure vessel and flows across the membrane surface, the water molecules are forced into and through the membrane envelopes, leaving the salt molecules behind. The desalted water passes through the membrane and emerges at low pressure, where it is collected in a tube and directed to one end of the pressure vessel. The concentrated salt stream that is rejected from flowing through the membrane continues to pass across the membrane surface, where it is collected separately. The concentrated salt stream retains about 55% of the total energy of the seawater stream that was originally fed to the membranes. To reduce the energy that is required to operate the reverse osmosis plant, the pressurized concentrated stream is piped into an energy recovery device. Here, up to 98% of the energy of the concentrated salt stream is transferred to an equal volume of the incoming seawater in an isobaric energy recovery device. The energy recovery device significantly reduces the plant's operating costs by recovering the concentrated salt stream energy and using it to pressurize 60% of the seawater that is fed to the membranes. The concentrated salt stream will have about a 60% higher salinity than the incoming seawater. The concentrated salt stream is sent back to the ocean through a brine disposal well, or a device known as a brine outfall. The brine outfall is situated in an area of significant ocean flow so that the salt levels are quickly returned to equilibrium with the ocean. The location for the outfall should contain no sensitive marine ecosystems. In a properly designed brine outfall, no noticeable increase in salinity can be detected at a distance of a few meters from the discharge. The pressurized seawater leaving the energy recovery device has its pressure boosted by a small booster pump so that it is at the same pressure as the seawater leaving the high-pressure pump. The boost is necessary as some pressure has been lost as the stream travels through the reverse osmosis system. Approximately 40% of the seawater that enters the system is converted to potable water during the reverse osmosis process. The potable water is further treated by adding calcium carbonate to improve the taste and bring the pH to the neutral range. Chlorine is also injected to provide disinfection properties as the water travels from the reverse osmosis plant through the distribution pipes to homes and businesses. When proper conservation of natural water resources is practiced, water reuse has been applied, and a water deficit still remains. Seawater reverse osmosis can offer a sustainable alternative. With good stewardship, it can provide life-sustaining water for coastal communities. Desalted water supplies, which are not susceptible to drought and other natural disasters, can provide a clean, safe potable water supply.